First, I thank you, the organizers, for giving me this opportunity to present you Pencello. So Pencello is a very early-stage company, and with a very excellent management team that we have. But it's an early stage, but we have a very, very high ambition. And you see and try to convince you during this 15-minute talk. This ambition is actually based on the 30 years of experience of my academic lab in the Mount Sinai Hospital in Toronto. And practically these two technologies that I'm going to present you, which made this company funded, is established and developed in that lab. So the two technologies that, as I said, it's funded the company was a few years ago we started thinking about what are the two major hurdles in cell therapy and regenerative medicine fields, and then we identified the two, which is cell therapy safety, and the second one is allogenic cell tolerance in an otherwise normal functioning immune system. So these are the two IPs that we filed. The first one is addressing the cell therapy safety, and we have a fail-safe technology. And the other one is building on this fail-safe technology. We started developing something which is intrinsically dangerous thing to do, hide cells from the immune system, and came up with a solution, a unique solution for inducing allogenic cell tolerance on a fail-safe cells. So practically, if you combine the two, if you have a fail-safe technology, fail-safe cell technology, which do not allow cells to develop cancer, for example, and combined with immunocloaking, the two together could actually bring us to the position where we can propose one single cell line for all humankind as a source of off-the-shelf therapeutic cell products, whatever implication is that going to be. So we were quite happy that our solution for safety of cells is recognized, and then we were able to publish this technology in Nature just a few months ago. And practically, this is the first solution where we can define the safety level of cell therapies in the number of cells needed for therapy, and also providing a genome editing solution with, according to our opinion, the best what we can do to implement or introduce a kill switch into the cells, which allows us to eliminate any bad behaving cells. So I don't want to spend too much with this because it's published, so that it's a cool paper to read, so that I highly recommend. But we had to come up with a solution, what we mean about fail-safe cells. And this is a definition that if we have an absolute external control on cell proliferation, this is how we call the cells fail-safe. Obviously, a single cell is not enough to treat disease, so that we have to produce a batch of cells, and we call this whole batch fail-safe, or fail-safe population of cells, if all the cells in the bag is fail-safe. So we have an absolute external control on their proliferation. So what we can tell the patient when the patient needs, let's say, 100 million cells or 10 billion cells to treat cardiomyocyte, we tell a number to the patient, and this is the number. The number of batches we can make to treat your disease, which this number allows you only one batch, which is not going to be fail-safe. And I tested this in my 9-year-old daughter, and then she could understand what then the fail-safe level is when I told this definition. So that was good. So that practically this allowed us also to think how to build a kill switch into the cells, which gives the biggest or the largest level of protection of the kill switch against all the dirty tricks of cells, which could eliminate a transgene. So I skipped this one, but I just tell you that this solution that we published in this paper allowed us to quantitate the safety in the function of number of cells needed for cell therapy. And let's see if it works. Nope. Okay, so pointer is not working, so that on the x-axis, you see the cell number needed for cell therapy from 100,000 cells to 10 billion cells. 10 billion cells what is you might need to treat myocardial infarct, and about 100,000 cells when ophthalmologists start talking about to treat blindness. So if you see that if we have a genome editing for the fail-safe kill switch, and we introduce only one system into the cells, the fail-safe level for treat the eye with 100,000 cells is 10,000. So you can actually treat 10,000 patients, and only one will get a non-fail-safe batch, which means that if something wrong happening, then we cannot eliminate those cells with the kill switch. So we have to do it by other means. So, but we also came up with a second level of a second generation solution, which is the green line there, which gives a really astronomical level of safety for all the cell therapies, including cardiomyocytes, where you are talking about 10 billion cells to inject into the heart. So with that fail-safe system, the fail-safe level is 1 million, which means that you can treat 1 million patients with cardiac infarct, and only one is going to get a non-fail-safe batch of cells. So I think it's quite acceptable level of safety. Just to show you here that how good control we have in cell growth with this system. So we created a tumor in the mouse with the fail-safe cells by injecting pluripotent cells under the skin subcutaneously. This is how you generate a teratoma in the mouse. And with the fail-safe system, so we're able to stop the tumor growth at the size when we want it, so around 500 cubic millimeters. We apply the product on the kill switch, and you see that this like small little bump, which is growing like crazy in the mouse, normally with normal cells, and going to kill the mouse in a few weeks. Now it's going to stop growing, and that remained stable, went into a dormancy for the whole life of the mouse, practically for a whole year. So having such a strong control on cell proliferation, so we were quite happy to think about what else we can do now, which is intrinsically dangerous thing to do if you have such a solution for the cell therapy safety. And this is where my favorite quote here: "The man is the only creature that dares to light a fire and live with it. The reason, because he alone has learned to put it out." So we consider the solution for safety is learning how to put the fire out. And this is where we went into another challenge, which is a kind of summarize the importance of it in this slide, which is kind of borrowed Mahendra Rao, who actually created a review about how the world is thinking about what cell type is going to be used, pluripotent cell type is going to be used for treating human. And the first column, you see that the idea of Yamanaka, when you are thinking about generating patient-derived iPS cells and change the iPS cells back into a therapeutic cell type and treat disease. We know the problem here. This is extremely expensive to do. Good advantage, however, is that it is a patient's own cells, it's autologous cells. The patient doesn't require immunosuppression. Even Japan is thinking about the second column, which is generating an iPS cell bank, which is carefully selected for HLA, homozygous HLA haplotype, and this bank can give the Japanese population about 80% good coverage of a good matching histocompatibility from the bank. This is less expensive and less time needed to create the therapy, but still quite expensive compared to the third one, which is you're thinking about, dreaming about a single cell line for all humankind to be treated. Obviously, the big problem with that is the need of serious immunosuppression in the patients, because these cells, most of the time, doesn't have histocompatibility match. So our ambition was, can we change the need of high level of immunosuppression to no immunosuppression, making the cells invisible for an otherwise functioning immune system? So here we have the solution. So we went not touching the HLA system, but actually identifying genes, immune-moderated genes on the cell surface, which allows us to hide the cells or cloak the cells from the immune system or protect the cells from any immune attack. So these are the 8 genes that we identified. We highly expressed these genes on the surface of the cells, and it did the job. So that this is just the first proof of principle experiment. In the mouse on the left pictures, you see then a black cell, which is not immunocloaked, just a white type cell, goes in the upper foremost into black mouse, isogenic. These cells expressing luciferase, you see that it's going to remain in the animal, totally accepted. No wonder it's isogenic cells, it should be accepted. But all the other different color recipients, like the white and the brown and the white again, which are allogenic, they reject the black cells injected under the skin. On the right side, however, it shows that the isogenic cells are okay, as I said, or as I showed on the left side, to stay in the mouse because it's isogenic, not rejected. But if you immunocloak the cells with this 8 transgene expression, these cells are also happily growing in an allogenic situation, black cells in the white mouse. So, and this is now the combination of the two technologies when you combine the fail-safe cells to the immunocloaked cells and show that this teratoma that we're generating from allogenic cells and stabilizing it with the fail-safe technology is staying in the mouse long-term for a year without any sign of immune rejection. So this is a very healthy tissue. We renamed the tissue now. We're not calling it teratoma. We just call it healthy, allogenic, friendly tissue, right? And this is what we can think that it could be also an important future application to treat disease. We can generate an allogenic tissue which are staying under the skin for long-term, not rejected. Could express different factors like missing factors, hormones, and so on. Treat endocrine diseases. Important note here that we have a proof of principle demonstration in the mouse. Obviously, we are moving in the system into the human. The challenge here, how to show long-term tolerance in the human without putting the cells into human. We are working on it, and we are working also to putting it into a non-human primate system. Good news is that these 8 transgenes are highly conserved between the mouse and human, so just because it's a functional ortholog, we expect the same what we can get in the mouse also happening in the human system. So here we are now, so that with these two technology, we are really in the position that we are, we can come up with a single cell line to treat all humankind, and then this single cell line could actually serve as a source of off-the-shelf therapeutic cell product of many, many implications that you can imagine. So, and the Pencello is now looking for partners with different implications in diabetes, arthritis, blindness, cancer, and many others that you can think about. And then with this, I would like to thank you to listening, but also to our local supporters in Toronto, the Center for Commercialization of Regenerative Medicine, CCRM, our institute, the Mount Sinai Hospital Research Institute, and also the Cranfield Foundation. Thank you very much.